This is Chapter 1 of the Instructor's video manual for cloning a fluorescent gene. In Chapter 1, we're going to get students familiarized with the tools and the vocabulary of a modern biotechnology laboratory. Chapter 1 is designed to get students used to two tools. The first is an air displacement pipette, which is used to transfer very, very small volumes of a reagent in the lab up to as little as 1/1000th of a milliliter from one tube to another. We're also going to be getting students used to using microcentrifuge tubes, which are the standard receptacles for reactions in biology. These tools take a little bit of getting used to before your students will be comfortable with them. That's why we created a supplementary video that gives tips and tricks for using pipettes and for utilizing microcentrifuge tubes. You should show this video to your students before sending them into the laboratory. To encourage teamwork and foster cooperation between students, we've designed CAFG so that two students will form a pair to do cloning a fluorescent gene. A pair of students will share a pipette, and two pairs, or 4 students, will be organized into a group which will share a common pool of reagents. Chapter 1 is split into 2 parts. In the first part of the lab protocol of Chapter 1, we just want to get students used to using the pipette and used to transferring volumes into and out of centrifuge tubes. We've provided 1 tube of phenol red, a dilute acid, and a dilute base for each group of 4 students. Each student will be responsible for mixing small amounts of phenol red, acid, and base, changing the color of the liquid in their microcentrifuge tube from red to yellow and back to red. If a student is incorrectly using the pipette or not mixing the liquids sufficiently, the color change won't happen, signaling that the student is doing something wrong and needs to refine his technique. In the second part of today's lab protocol, each group of 4 students will be separating out enough of the reagents for them to use for the remainder of the lab course. On the instructor's lab bench, you should have separated and thawed all of the reagents except the enzymes that the students are going to need for the rest of the lab course. In each tube that we've provided you, there's enough reagents to be split between 12 students. That means that 3 groups will come up and split off enough for their group of 4 from the 1 tube for a group of 12. This is called aliquoting, and we do it all the time in modern biotechnology labs. Once the students have created aliquots for all of the reagents that they're going to need for their group of 4, their group should put all of their tubes in a plastic bag or some other container, label it with their name and initials, and put it in the freezer, where it can be stored until you're ready to use those reagents.